Reinhard, I want to thank you for really doing all the work to put this together. And so it's a real opportunity. I'm very grateful to be here and to look out at all of you, and then, of course, the view out in the background. Okay, so I want to talk about testing modularity. And so I want to start with the obvious question. I throw something up, is that a module, right? And as we've heard 2 talks at least, what is a module? I have absolutely no idea, all right? But we've seen this before. You know, what is intelligence? And the way that seems to have been decided is by the Turing test. So if we don't know how to define something, let's throw up some ideas and then we can see whether we believe that we have a module, okay? So I think I'm kind of replicating some of the things we've already heard today, and that is properties that a module should have, all right? They don't, and I don't know the mix of these properties that are necessary for us to say this is a module, all right? But I do think all 3 of these properties are necessary. The first one is it really should do something, okay, or else who cares about it, all right? So there's a functional aspect to it. As we've heard when we were talking about, you know, Artemis, right? You want to make sure that you put them together and you send it out to space and it still works, all right? So there has to be some amount of robustness to it. But at the same time, it's not just supposed to be a rock that sits there. You got to be able to put inputs to it and have it actually work. And in many cases, we actually want that different inputs give us different outputs. So there has to be some amount of sensitivity to it. In addition, if we think about it, and that's kind of where I'm spending most of my time thinking about modularity from the biological sense, there should be some evolutionary aspect to it. And so you know, there isn't a creator that just says, here's the module. You have to be able to work your way to it, all right? So there has to be some malleability to these objects, all right? So what I'd like to do is to say, okay, here's some questions. We should, if somebody gives me something and says, this is a module, we should test it against these questions. And I'd like to be able to quantify, in some sense, answers to each of these questions. And then, okay, if it has the right kind of mix, then we call it a module. And if it doesn't, then we say it's not, all right? So I'm trying to punt on the question of a definition. But if you agree with me that these are kind of the 3 things that are necessary, then they actually take on different roles kind of in the modeling. The first one is really a question about dynamics. From my perspective, you have an input, you're supposed to get an output, there's something process that went on, so you've got to understand dynamics. The second and third are somehow a little bit different. They talk about this object you have sitting inside a bigger universe, and you need to understand how that universe impinges on what's going on, all right? And so I'm really going to kind of give this talk in 2 parts. I'm going to address the question of the universe that we want to think about when we do this modularity, and then I'm going to talk about what dynamics happens within this universe, and then maybe the mix of these would allow us to address these 3 questions, all right? Now, I've got a lot of things I want to say, but I don't care if I finish. I'd rather have questions along the way, okay? Yes?
I'll start then.
Yeah.
So I'm a little bit puzzled by the second statement you had for your criteria. You said that it had to be robust with respect to the inputs. So you're thinking like it has to be like a continuous function? Is that your thinking? Because robustness a lot of times in engineering will mean something can change inside my system and I'll still produce the right output.
So I'm thinking both, internally and externally.
Okay. So,
yes, right. I'm trying to be as broad as possible. But yes, that's a very, please, these kind of questions, yes.
I also wanted a quick clarification about number 3.
Yeah.
Is that saying something about it, the function itself should change based on external inputs, or is it just saying the function should be non-trivial?
I think I want to, okay, let's, yes to the first one, all right? I mean, I think we often want to have a switch, and a switch should be a module, right? You want an on and off state. Maybe at this point it should be on, and at this point it should be off. Okay, so that means that the module itself has to be able to produce different functions depending on different inputs. But the other part also, if we think about getting to a module via evolution, you have to be able to tune it a little way to get somewhere, right? And then you might, if you're a control theorist, say, oh, I'd like to, if you're a synthetic biologist, you might say, how would I change it in order to make it perform differently? Okay?
So I would want those three things for any model, not just for the modules. The things you listed.
But it's hard to distinguish between a module and a model, right? Yes, I agree, but that's why I'm not trying to give you a definition. We can check this and then say, well, that's a good model, or we really want to think of it as a module because I'd like to be able to put it into the context of a bigger, yet bigger system. I think the hierarchies is really important. Yes.
Just to make things interesting, so would these three rules, a biological clock would not be a module?
Oh, why not?
Because it doesn't change. You want it to be as steady as possible.
Okay, so but there's going to be lots of inputs that come in, right? So I fly to Europe, and my circadian rhythm, right?
Yeah, but what happens if you have a clock that has to be fixed?
Right. It must be someone.
Okay, then, I mean, it has to be fixed, but there are going to be places where it breaks, and then we need to understand how much it breaks, right? Right? I'm trying to quantify these, and I don't think any single one of these is sufficient, and I don't really know what the mix is. I think the individual, when they make a model of the system, they would want all three of these, to understand all three of these properties, and then kind of, if it's part of a bigger system, I'd like to understand how much these properties are preserved as we put it into the bigger system. Okay, and there was one more question. Yes.
So I have a question about number 3. It says the function change depends on the inputs. So can I think of like there is some threshold level to which it is robust, and then after that, if it goes beyond that, then it changes the outputs? That is one question. And the second question is, if there is a notion of typed inputs are itself classified into types, and with that type of inputs, there is a desired function of type 1, and with that type, it is robust in that way. Again, if we change the total type, then the desired function is something.
Yeah, sure. No problem. Yes to both of those, right? I'm trying to be very vague, right? I'm going to try to build a modeling framework for you. Okay, so I'm going to try to talk about a framework in which we could try to address those questions. Do I have all the answers? Absolutely not. All right, but I'm going to pretend I do. Okay. Okay, so I'm going to go through a bunch of stuff, and please stop me with the questions. Right? You know, I really don't need to get through all this. So let me start with a biological question. All right, because there's a very broad audience here. I'm going to try to give a math talk. And I'm going to try to introduce ideas that maybe don't make an obvious connection to, say, biology. So I'm going to try to hop back and forth, okay? It's up to you to try to make sure that I'm not hopping too wildly, okay? So here's my biological question. And this is a complicated question. I do not have the answer whether this should be a module, but I'm going to try to explain the math, and so I'm going to give you very, very simple examples. So I'm also trying to convince you that maybe you can actually do something with what I'm talking about, okay? So this has to do with yeast cells. There's something called the mitotic cycle, which is, if you're a pretend biologist like I am, then that's what cell cycles are all about. The DNA replicates, the nucleus comes apart into two of them, the cell comes apart, and you have two cells. And that's what I thought replication was all about. But apparently there's an endocycle where the DNA separates, but that's it. And then you go through that cycle again and again, all right? So this is, if you're a biologist, maybe this means something to you. This is coming from Well, those are the papers where you see those pictures from. And so the question is this an appropriate module for these two types of phenotypes? All right, one is the cell cycle going through what I would think is normal division, and the other is endocycle. All right. So that, and I do not know the answer to this. Okay. I will talk about some aspects that we've been able to do here, answering parts of the questions, but there are other questions that we're still trying to address.
Can I ask a question?
Yeah.
Slide? Are you saying that that system, this diagram here at the bottom, controls both the cell cycle and the DNA division cycle, the endocycle?
Yeah, that's kind of the claim, right? And here, I mean, there's, again, Steve, this is work with Steve Haas. If he were here, then I would be directing all biological questions to him. But I have co-authors like Thomas Gideon, who understands the biology much better than I do. So I'm going to divert. He has to be my substitute, Steve Haas. All right. So if you go to a standard biology textbook, there is a gene regulatory network for the cell cycle. My understanding is part of that's embedded in here. So this module might be kind of an overlap of things going on. So it's not quite as well divided as the electrical engineering part, right? But maybe we want to allow that for modularity too.
So, Konstantin, when you say module, do you mean this is a part of some bigger?
Well, it's part of the cell, right? There's lots of stuff going on, right? And there's two different phenotypes going on. And I'd like to have something that's well-defined that allows me to see what's going on, all right? Yeah.
So when we ask this question, like, is this an appropriate module? So you have told me what the function is, right?
Yeah.
But like, don't we also need to also mention what is the external inputs and what they can change?
I will get to that. Yes.
I just want to clarify the endocycle a little bit. So instead of DNA divide, it's actually replicate. So like, if you consider like your input is some type of DNA or whatever, like it accumulates.
Yes. And apparently that's what we're supposed to be seeing in these pictures, right? Yes. So it's really going, and I think Steve said they get up to 8 replications.
I mean, I think the greens are the markers for the DNA copies. So I call it whatever the.
Yeah.
So it's like,
I think the green is actually one of the protein. The blue is the Hartree staining for the DNA.
Again.
The blue is a Hartree staining for the DNA. It's a it stains the double-stranded DNA. And the green one is actually a certain type of, probably like, fraction staining for something.
it's this, it's an, what is the center which takes the, which.
Yeah, the centromeres, but it's not the DNA itself.
Exactly.
All right. So you can see my level of biology. All right. Okay, let me keep going. Okay, that's what I would like to get some answer to, a partial answer. I do not have a complete answer, right? Okay, so I want to talk about universe as a module of models, right? Because for 2 and 3, I need to be able to talk about what's going on outside of the module that I have. Okay, so I'm going to start with an interaction network. And what I mean by this is that, 1 influences 2, 2 influences 1. And I'm not telling you whether it's upregulating or downregulating at this point. I'm just giving you a network diagram. Who talks to whom? I think that's about as crude a input as I can get. And I want to use this picture to organize all possible dynamics, right? In a way that I can compute with, okay? So I'm going to start with local information. And I'm going to say, how does an individual node in that big diagram process and relay information? We've already heard one idea, and this is an idea that I think is very important if one really wants to talk about a universe of what's going on, and that's this Boolean idea, right? That things are kind of on and off. And so I'm going to build a combinatorial model on which everything is going to rest, because I want to be able to really search everything. Okay, a little bit of notation. I only care about the number of outputs, and I only care about the number of inputs for what I'm going to do right now. Okay, I have to build a parameter space. All right, I'm going to tell you there's lots of possible dynamics. I'm not telling you what the dynamics is yet. I'm just trying to say here's the universe that captures all the dynamics. So I have to parameterize it. The first part of the parameterization is something we call an order parameter. And this is just looking at these out edges. I've just labeled them. There are no numbers here or anything. I've just given you a labeling. And I'm ordering those labels. So if I have, Tn out edges, and then there's Tn factorial different ways to order things. And this is supposed to somehow measure the relative sensitivity of downstream nodes to the information that's coming from node N. That's the intuition behind this ordering. But there's all this information coming from upstream, and somehow that has to be processed. And here's kind of where the Boolean kind of ideas come through. I'm going to eventually, because I want to keep things simple, we can make it more complicated if we need to, but I want to think about, kind of monotone information being passed across. And so I'm going to encode this incoming information via, in a monotone fashion. And so what I look at is off, on, and there's Sn coming in, so I'm going to take a product of that. So each one can have an off and an on. And it's going to come out and give me a bunch of outputs that are going to be handed off to the outputs here. So this is just a Boolean function, but I want it to be a monotone Boolean function, right? 0 is less than 1. If you think of a product of zeros and ones, then you can talk about two of these Boolean vectors, one being less than the other, if coordinate-wise you have less than or equal to in each coordinate, all right? Okay, yes?
Do you have a feeling about how you have limited the space of functions that will abide by that property? Not all functions would. You would not necessarily always have monotonicity.
Okay, so if I don't have monotonicity, I'm going to put in more edges.
Okay, all right.
Okay.
So,
Okay, but I don't want to, that just makes things even more complicated,
all
right? And then the question is, kind of, can you really think about it in terms of these individual vectors? And then there's this question of Hilbert that I think he posed as, is there really ever a three-dimensional function that really is a function of three variables? And Kolmogorov and Arnold proved that no, that's not true. You can always write functions as complicated functions as functions of one variable. Now, the functions you make are really, really terrible. Okay, but conceptually, I think, big question if there are any, people who know this theory well, it suggests that maybe everything can be encoded into something like this. A big question mark there, right? Okay. So a factor graph. So I have node n, and I'm going to make something I call a factor graph, which is a product of these parameters and these parameters. Okay, so for every node, I build this factor graph. It's huge, right? Because I've already put, this grows very fast and this grows very fast. And I want to organize all this thing. So those are the vertices, and I'd like to have edges, and I'd like to be able to say, well, this node is close to this node in parameter space, and that's going to happen if I flip just one order. Now, I put this is supposed to look much lighter in color than that because I haven't given you enough information. I'll come back to that in a moment, right? So there's going to be this thing called a parameter graph. You have your network, your directed graph. You go to every node, you look at the factor graph of that, and then you take the product of all the factor graphs, and that is the universe. Okay? So there's this directed graph that not directed, this graph with nodes and edges that tells you what the universe looks like, And for this particular example, that universe has exactly 51,732,000,000,000 nodes in it. So it's absolutely enormous, yes. The universe,
For patterns or connections.
yes.
Ah.
Right, it's all those, I gave you the parameters. I told you how to make a parameter, a factor graph for each one. I take the product of that, and those are all the possible, that's the universe, that graph.
All possible graphs.
Yeah. And no, there's one graph, absolutely huge graph. I haven't told you the dynamics. I need to do that. So for every node in this huge graph that I made, with this many nodes in it, for every one of these nodes, I'm going to give you a dynamical system. Yeah, okay. Other questions? Okay. Let's talk about modeling dynamics. And I'm very interested in making sure that these models actually could be carried over to something maybe more continuous, all right? Because I think a lot of our modeling is, you have a certain number of proteins, and the way the number of proteins changes should roughly change continuously. Obviously, that's wrong because you got a unit protein. But on a large scale, maybe we want to do a little bit of chemistry, and so it'd be nice to think that there was an ODE model that might fit to this system, okay? So I'm going to do it via a very, very simple example. That's about the simplest example I could think of, a one-node network with a self-edge, okay? Notice I put an arrow in here because I'm going to now talk about dynamics, and I'm telling you this is upregulating. So one upregulates itself in a monotone fashion. Okay, so there's going to be now a real number that we could associate to it, okay? And here are kind of assumptions that I'm going to make, because I'd like my dynamics to be able to match these kinds of assumptions. And I break them up into three groups. One is what I think of as biophysical. So changes in the number in this quantity are going to be, well, a sum of the decay rates and the creation, all right? This actually is a strong assumption because I'm going to put a plus, all right? Okay, so I've separated creation and destruction. So that would say, look, if I was trying to write down a form, a functional form for how things are changing, I'd have something decay, which should be proportional to the amount that I have, and creation. That's too complicated for today's talk.
What is X here?
X is the quantity that I'm going to associate with this node. Maybe it's a protein.
X1 or X?
X1. X1.
What is X? Not XX.
Oh, yes, thank you. If I go to a bigger network, then there's lots of things that could be impinging on it. Yes. So yes, thank you. Please keep asking questions. All right, yes. This is looking at the entire network, and this is the decay for X1. Yes.
Question?
This case F1.
Why only the decay function is proportional to the amount and not the creation function?
I didn't say it wasn't. It's just
Okay,
that,
just to make it more generic.
Yeah. Okay, obviously, and I think this is a big point, I do not know the function, and this is typically a very expensive computation to do, right? Not computation, experiments to do. You've got to go out and figure out what actually is happening, and, so I would like to work in a universe where I don't need to know that a priori. Yes?
Are you assuming that G and F do not change over time?
For what I'm doing right now, yes.
Okay.
I'm happy to talk to you about if we want to put time changes in there, okay? And then it becomes a question of whether you want to think about this from a control theoretic point of view or you want to think about it from a dynamical systems point of view as to how you want to think about those changing with respect to time, okay? So I don't know F and I don't know gamma, but I'm going to assume that the X's are positive. They're supposed to be quantities. I'm going to assume that decay really decays. I'm going to assume that creation really creates. And I'm going to assume that there's a limit to the amount of creation you can make, right? There's only so much protein you can produce, a rate at which you can produce protein. The biostatistical is, you know, I put down a network for you. Where do we get those arrows in these networks? You do some experiments and you do some statistics on the experiment, and then you say, oh, node A talks to node B. So that's what I'm telling you there. And the final one is, and this goes back to this monotonicity. When you see me put an arrow, I'm going to assume that Fn is monotone increasing in terms of Xm, and Fn is going to be monotone decreasing with respect to Xm.
May I ask about
Yeah.
Your categorization of assumption 2 being statistical?
Yeah.
Why is that a statistical assumption?
Because I think that there is my understanding is there's very few places where you actually understand the biochemistry of an interaction. Okay. There's a lot of this throughput experiments that say, oh, here's the network that goes with what's been done. And you haven't actually followed the exact proteins and seen exactly how they bind and understand the binding of it. And
So there's a probability associated with how.
right. I'm just calling this a biostatistical because I see that statistics gets used a lot to determine this. Okay. Okay. Now let's finally talk about encoding the dynamics. There are a bunch of steps to this. Again, stop me as I'm going through here. So I have there's just a single variable. So the phase space, whatever it should be one-dimensional. But I don't want to deal with numbers because numbers are way too hard to work with. So I'm going to make some abstract thing called a cell complex. If you're an algebraic topologist, you know what I'm talking about. But what this really is there's a one-dimensional cell, there's another one-dimensional cell, and there are vertices. I drew them this way because this one-dimensional cell has these two vertices as boundaries, and this one-dimensional cell has those two vertices as boundaries that can be encoded algebraically. That's the way we think about it. So, but it's easier for us to think of a geometric picture. So what's stored here are just 5 objects. 2 of them labeled as one-dimensional, 3 of them is labeled as zero-dimensional, and there's a little bit of algebra that tells you that if you're going to draw a picture, you draw a picture like that. Okay? There's no numbers here. Now, here's my network. There's an order parameter. There's only one out edge, so the order parameter is just a single object, so it's hard to see orders here. We'll get to that later. And there's the logic parameters, right? Inputs to outputs. Oh, S1 is 1 and T1 is 1. There's one input, there's one output. So we're really looking now at these monotone functions from 0, 1 to 0, 1. And we're going to make them monotone increasing just because I want to organize things. So this is a monotone increasing function. 0 goes to 0, 1 goes, that's kind of a boring monotone function. 1 is bigger than 0, but 1 can be sent to 0, right? It's a constant function, it's monotone. Here's what you'd really think about as a monotone function, and that's also a monotone function. What is not allowed, because I want it to be monotone increasing, is something where 0 gets sent to 1 and 1 gets sent to 0, because the low thing got sent high and the high thing got sent low. So I'm not allowed to look at that function. All right? Okay? Okay, there are 3 functions. There's one order parameter. 1 times 3 is 3. So I must have a parameter graph that has 3 nodes. Yes?
Don't your first and third functions violate your, yeah, your first and third functions violate your biostatistics assumptions, don't they? Because the output doesn't depend on the input.
Well,
or
it doesn't violate it, right? I'm telling you that A impacts B, okay? But maybe that impact is so small that I can't measure it.
Okay, fair enough.
Right? I'm a mathematician. I always like to keep track of ridiculous cases, okay? But it's very important if I want to look at the space of all possibilities, because maybe one of those edges gets broken. And Now I'm allowed that to have that into my model. Okay, so I'm going to have 3 nodes in my parameter graph. So I have to have 3 different types of dynamics. Over here is where I'm just putting this in kind of to try to keep honest with the assumptions I made. This was this assumption that F1 is a function of X1 that I get from the graph. I mean, it's a triviality at this point, but this is where it's these logic functions where you would see that statistical, the biostatistical information coming in. I'm going to say, well, there's some special prop points in my dynamics. 0, for example, there's no protein. That's kind of special. Infinity, or something really, really large, is another special case. And so I will label those. And let's use the assumptions. So I'm assuming that production is always positive. So if X is 0, this term's gone. So this thing should be producing the protein. And I'll put a plus sign to indicate that. So very low concentrations, if you're looking at what happens at this endpoint, I'm saying things are going to increase. Let's go out to infinity. I told you that this thing's supposed to be bounded. Huge decay, some bound. So with respect to high levels of whatever you have, you should see things decreasing. Okay. That order parameter now appears in the dynamics. I'm just labeling this vertex via this threshold. If there had been 2 out edges here, I would have put 2 vertices here with an extra edge. Okay, you'll see that later. So here is this monotone function, 0, 0, and I'm going to put the threshold above that. That kind of says, oh, at this point, I don't see the impact of the incoming edges. All right, this is a different way of thinking about your question. And so, well, things are just still decaying because I can't see that the creation is having an impact. Here's the monotone function. Low is low and high is high. And so as I pass through this threshold, I see a change in the dynamics. And finally, at this point, I'm telling you everything's always on. So that threshold is kind of always below what we're seeing happening. Okay? I want to emphasize I've given you words, but this is pure combinatorics. All right? I'm just telling you how to label these cell complexes as a function of these input parameters that I've given. So again, this place right here where these arrows are going in here this way is because of this. If I'd had a blunt arrow, I would set everything up the same way, but I'd have to flip the signs. So that's how I put negative, I put repression into the system. Okay, here I finally get to show you what I mean by flipping, right? I flipped an order relation, so I put an edge. I flip an order relation, so I put an edge. So this here is the parameter graph for the 1-node network. There's 3 types of dynamics, and some of those dynamics are closer to others. Yes?
That may seem like a technicality, but I don't understand. So in the second case, right?
Yes.
Why do you have plus, minus, plus, minus? Why is it not plus, plus, minus, minus? In the sense that at the order parameter, you're now getting.
If I want to think about it from a say a differential equations point of view, I'm kind of over here, and I'm thinking of putting that this quantity is theta. All right, and so now I'm telling you that if I'm at a low level, then this is in here. This is negative. This dominates this, and over here, this dominates that. Okay, and that's why it becomes a plus. Does that make sense?
I don't see how you can get this non-monotonic behavior in there, that the quantity is increasing,
well, okay.
and then it can increase.
This is boundary conditions, right? And this is saying F is increasing. So as you increase, the rate of production gets larger and larger. At this point, it gets large enough that over here at these low levels, I see it decreasing. But when I go to these levels, I see it increasing.
Two terms.
Other questions? Okay, we call this a wall labeling. So this parameter graph that we have, the nodes, every node in the parameter graph corresponds to a particular wall labeling. And it's from the wall labeling that I'm going to produce the dynamics. Now, I told you that I build this complex, but this complex is not rich enough for us to do measurements. I mean, this obviously is a place of interest, but if I just think about it as a zero-dimensional object, it means I can't kind of go in there and do experiments around that. So I would like everything of interest to be a one-dimensional interval. So I can make measurements and I can have errors and I can still talk about what I'm doing. So we create something that's a slightly bigger complex where every vertex, every cell in the original complex becomes a top dimensional cell in this blow-up complex. And then I'm going to build my state transition graph based on those top dimensional cells in the blow-up complex. And so what we're doing here, here's my wall labeling. So this vertex became this cell. What happens when I look at how this cell looks at this cell? I'm increasing. And so I'm just going to put an arrow from there to there. That's this ODE, this continuity kind of assumption. I have a plus sign here. Why don't I jump all the way over to there? Because that would be a huge jump. That would be something that's discontinuous. Okay, so this guy gets moved here. This guy gets moved here.
That first column that you're just talking about, that's your last function, right? The last function on the previous slide?
This is his wall labeling.
Right, but on the previous slide, that corresponded to where everything's being sent to 1.
Yes.
Zero.
Yes. Everything was sent to 0. Right? Which did I get?
Switch.
Oh, I switched the orders. Sorry. Yes, this was everything 0. Everybody, of course, mentally can easily. Sorry. Okay. Yeah, I know why that happened, but sorry, I apologize. Okay. I'm going to say that this cell should get mapped to that cell. That's this monotonicity condition. Things are just increasing as I go across. So if this is sent to the right and this is sent to the right, then everything should get sent to the right. Okay, so that's the monotonicity. And I keep going through, except note here I'm coming in and here I'm coming in. Oh, there's something that's going to get stuck there. So I'm going to put a self loop. Okay. Okay, so that's what happens if you do it. Okay, so in one dimension, it's really easy to make this state transition graph. If we go to higher dimensions, it's hard. Okay, but fortunately, Bernardo, and I don't even know where he's sitting, he's there. So that was part of his thesis. So people can go talk to him about that. Okay, so let's now try to characterize the dynamics. And now I'm going to come back to kind of the stuff that Reinert was talking about. Yes?
So you said dynamics. So now, does that mean like so when you say dynamics, you're trying to figure out.
I have a state transition graph. That's what I mean by dynamics at this point.
But the state transition graph is like describing the dynamics of X, the concentration of X1? Or
it's purely combinatorial at this point. It's just, cells getting mapped to cells.
Okay. And what happens if there are 2 out edges? Like, so you will put 2, like it will be a 3 1 simplices attached to one another, right?
Yes, but the simplices will be organized by the thresholds. Okay, let's talk about that in detail later.
There's non-determinism, right? The middle cell goes to both directions. Oh, sorry. In the last picture you had, the middle cell goes to 2, right?
Yes. Yes, was that your question?
No, I was just curious what the state transition graph was describing.
Okay, it's just a map that says this cell goes to that cell. It's purely combinatorial at this point.
You go back to the previous slide.
Sure.
So is it okay to interpret that as that's telling us how the system transitions from thresholds, threshold endpoints, and open intervals? That's
Yes. Yeah. Yes. That's yes. Okay. And making up these functions so that I can later do what I'm going to talk about is non-trivial. That's why you have to go talk to Bernardo about it. Okay. So this is where I'm sweeping a lot of stuff under the carpet, how to do this in higher dimensions. But it's well defined, all right? Or there are a variety of definitions one can impose that are going to work. But I'd like to characterize the dynamics. You know, when you go to a network of interest, the cell complex is enormous, and nobody wants to look at all these arrows, okay? So instead, what I'm going to do is I'm going to say, let's look at the recurrent part. So this cell went to itself. This cell went to itself, this cell went to itself, this cell went to itself. And let's keep track of all these recurrent pieces. And here everything kind of ends up at this place. Over here, this recurrent piece, if you leave it, you either end up here or you end up here. All right? So I've done something like the strongly connected components, but I'm really only keeping track of the strongly connected components that have recurrence within them. And then how you get from these recurrent states to another recurrent state. Okay? Okay, so this is called a Morse graph. So you have your state transition graph, you do a reduction to the Morse graph, you end up with a partially ordered set. The vertices identify the pulsal recurrent dynamics, and the edges identify gradient-like dynamics. So minimal objects in the Morse graph are kind of the stable objects that you could hope to see easily from an experiment. Can this be a useful module? I'm going to say yes. Here are the three. I've now labeled the parameter regions as the nodes in the parameter graph as R0, R1, R2. Notice these are kind of the same. So if I kind of think of these as equilibrium points, then this equilibrium point kind of gets carried to this equilibrium point, and this equilibrium point gets carried to that equilibrium point, and this kind of appeared out of nowhere. Right? So maybe I conceptually could think of a picture like this, right? And that's starting to look like a hysteresis curve, right? So it's purely combinatorial, but it starts looking like a hysteresis diagram. Okay. If you want to do this for large systems, okay, you've got to use, but the computer scientists have solved this, and Reinhard already talked about it. They're very fast algorithms for finding these Morse graphs for even very large graphs. Okay, so this can be done. And let's go back to my biological question. So let's try to answer that question now. So there's the wild type experiments that we get from Steve Haas. How am I going to address this? Well, I go and identify the parameter graph. I already told you what that was for this system. This actually contains all possible subnetworks, the way I set it up. Let's just sit and look at those parameter regions where every edge actually plays a role. Okay, so if I do that, then I've got a much smaller graph, only 1 billion. And what I can do is I can compute the Morse graphs for each of these billion things. The computations are Boolean-like, so they're very, very fast. So you can do it. And if you want to know how to do it, ask Marcio Gamero, who's there. What we find out is that about 2% of these essential vertices will reproduce oscillations, and those oscillations will match the orders in which genes turn on and off. Okay, endocycling. What happens in endocycling? You go and you kill CLIB2, knockout experiment. Our assumption is that the parameters for the rest of the graph aren't changed. So I'm still at those parameter nodes, except I'm only looking at parameter nodes where this was wiped out. Okay, ask the same question. Run the Morse graphs for each of those that you had and ask, do you see a minimal node in the Morse graph that's oscillating and matches the experimental data? And the answer is that happens for about 0.6% of it. So out of this billion regions that we've looked at, about 0.6 of them, that's still a large number, will produce both systems, both experimental outputs. So there's a certain amount of robustness to it. I know what the universe is. Another set of experiments that we're going to conduct, and we're developing software to do this, would be to understand where it fails and how does it fail. Yes?
You said robustness, you just don't have enough data to pin down that last 0.6%.
How are these? So the answer is yes and no. Let me give you, okay. I think that's the wrong question to ask, right? Because how are these experiments done? You take a yeast cell, you grow up, what, 10 to the 12th yeast cell? I don't know how many are in a chemostat, 10 to the 9th, 10 to the 12th. There are lots of, every time it replicates, there's a mutation that's happening. It might be very small, but maybe what really happens is that you really have a distribution.
Or you have a moving target.
Right? And this, I've told you what the universe is. So maybe I can think about where I am in the universe.
0.6%, that's the moving target. So those are all the moving targets.
Yeah. Maybe. But
I can understand one thing. Your measurements are continuous, not Boolean.
We're only looking at who comes on. We want on a cycle. We know everything has to oscillate. And then we say, okay, when does this guy, this is off. It was off before this was off.
Oh.
So we're really doing Boolean measurements.
Oh.
Okay. So give me 2 minutes. Okay. One, if we have the recurrent dynamics, I would really like to do more than just tell you oscillation or fixed points. This is where algebraic topology can come into play. So we can compute all the algebraic topology. That's why part of why I use a cell complex. And the algebraic topology can tell me, there are a bunch of theorems out there that say, oh, if you've computed this algebraic topological quantity, then you know something about the dynamics. All right. So this is what would happen there. And I want to go, I'm going to skip, this is a technicality, how we go from the algebraic topology to the differential equations. But what I'm claiming is that if this is my regulatory network, notice there's two out edges, so I get two thetas when I build my cell complex. All right, so those are all the thetas that are sitting in there. And I do my blow-up complex. And now I can do all my calculations, and I do those calculations with a particular choice of the state transition graph. And this is what I get for the Morse graph with the Conley indices: stable fixed point index, stable fixed point index, unstable fixed point index. What does this have to do with differential equations? Well, if you were doing Hill functions as your model, and you chose these particular parameters, then Bernardo would tell you and Marcio would tell you how to take this cell complex, embed it into the phase space for the Hill function differential equation model. All you need to do is kind of check transversality along these edges, and if that's satisfied, then I've already solved the differential equation for you because I did the algebraic topology. All right? So really kind of what we're doing here is setting up essentially automated the process of doing phase plane analysis, but for n dimensions. Now, to make the pictures easy, we chose very high Hill function. But there's a paper that should appear very soon in SciAds where we actually talk, and this is with Shane Kepley and Elena Coelho, about what happens as you bring this down. How should you understand that in the context of the full universe? And I think I'm out of time, so I will stop here. Okay.
Oh, I really wish I understood more of what you said.
Well, we have a week to talk.
And look, but just a couple of thoughts for you. A lot of what you're doing at the first part related very directly to the classical approaches people derive differential equations from chemical reaction networks. Very, very much analogous. So I don't know to what extent you're familiar with those techniques, but it might be interesting to overlay that. The second observation is that some of your approaches to dealing with, you have this Boolean stuff that's basically defining, portions of state space, and you've also got some continuous functions. And it reminded me of the way electrical circuits are analyzed in the presence of really non-continuous elements like transistors. And you're breaking it down to different parts. So I think there are a lot of sort of analogous techniques in there that might be interesting to intersect with.
We should talk about that. And let me kind of take what you said and twist it to my advantage.
That's fine, because I want to understand
Okay.
it.
So I think what we have, and this is joint work with Tomas and Bill and Marcio and people, I think we have a modeling framework where we actually know where the monotone Boolean networks sit within this. And the reason I kept you all late from lunch to show you the ODE is to say that we actually know how to go from what we've done to continuous models.
Okay, that's cool.
And if you start with the ODE, I think you're lost because you've kind of got, and you do simulations, you're locked into that particular nonlinearity. Here I'm saying, let's first understand what kind of dynamics we could see, and then I can tell you whether I can map it into a particular system of ODEs, right? And of course, it's much cheaper to compute than the ODEs. The ODEs give you much more precise information, but if you don't know the exact nonlinearities, sorry.
I think somebody there.
Oh, sorry.
Very quick question. When you map to the ODE, does it only you get exactly those three steady state only for those particular Hill function or any Hill function, any parameter?
So the mapping is actually extremely general. The only thing I need to know is kind of some transversality conditions with respect to certain subsets. So there's actually a huge family of ODEs that I've actually solved for every time I've done it. Question is, and this is partly why I mentioned these results of Kolmogorov and Arnold. The question is, can every ODE be modeled through exactly the framework that we've talked about? I would not be surprised if there's a counterexample. Marcio is more confident. He thinks we can do it. But there's, it might be that every single ODE can be modeled through this. I don't know. It's an open question. Yeah. Sorry. Yes.
So the way I'm thinking about your framework is it gives all possible range of parameters for dynamic features, signatures, you could say. Like in the positive self loop, you have like the single fixed point and then the bistable. But I was thinking like that could be like one model component. Like, have you considered, like, if I put together two of these positive self loops, would I get a similar product on the Morse graph? Like,
so Tomas has been doing a lot of work on that. I think a lot of negative results, right? Bill has been trying to do this from a more categorical perspective. And so he started, if it's fair to say, looking at products of systems, right? And trying to understand if there's a general framework for that.
Because from your initial list, I think composability is what's missing, right? So you're not considering like composing two things, right?
Right, but that is, you have to talk to Tomas. He's done a lot of work on what happens when you try to put two systems together. Is that a fair? Yeah.
I think, in the framework which Reinhard was talking about this morning, when I have things which are feeding into each other, then you can merge it with this one and get some results. But if I have two modules which are talking to each other in a circle, yeah, that's not going to work.
Well, I wouldn't say it's not going to work. We build a bigger universe, and then we have a way to study that. Yeah. Okay. I always try to be positive.
I actually have a biologic question.
Sure.
So this second truncated cycle, does it happen naturally for a certain environment, or is it a knockout phenotype?
It does happen naturally. Like you have these cells, I forgot the name, but you have a cell which have multiple.
Sorry, can you use the mic to answer? I do want to hear that answer, please.
Right there. She wants to answer it.
I don't want to answer. I want to hear your answer. Sorry.
So I don't remember all the names, but there are cells in even our human body, or when you have a injury, after injury, you have a multi-nucleated cells, which naturally evolve to build the chemistry faster, and then they disappear, right? So this actually happens naturally, even I think in human bodies, by many animals, and certainly yeast cells. So the knockout is just a model to do it, but it actually does occur naturally.
Skeletal muscle.
Yeah.
Yes.
I have a question. So when you are using the empirical data to strip where this is into like 1% or 0.6%, is that necessary that you actually know all the components to generate empirical data to this step? If this step can be omitted, what if you don't know like all the,
I don't know. Good question. We haven't tried to do experiments on that. All right. But let's talk about that this week.
Just to clarify, lunch is not until 12:15. The idea was the last 30 minutes was going to be discussion about all the talks this morning, but that's okay.
A very, very basic question. When you put up that first thing and ask, is this a module? Eventually, have you answered that question? It seems that structurally that you have demonstrated this particular structure that you have has two fates, right? So are you saying that the module is that structure and that it exhibits two kinds of state, or are you saying each of those states is a module?
Okay, so I was honest and I said I cannot answer that question even for this particular system. What we did show is that this network does match the questions that Steve was asking. So that's kind of number one. So it does do the function we want. We kind of understand in this universe that there's a large number of parameters at which this functions. So it has a certain amount of robustness. It doesn't, we don't have to be really, really tuned in terribly tightly and it'll still work. That third question, however, was kind of what happens if you walk away from this particular system, right? One thing that we would like to do is to try to understand what happens if you, instead of looking at only the essential, you take one step away in the parameter graph. So that's like losing an edge, and ask, can we still replicate this, right? That might suggest that there would be a smaller module that would work. But if it only works for a few nodes, then maybe you'd say, oh, that's not robust enough for me to think about this as the appropriate system. Maybe, and this now becomes, even more intractable, maybe I go into a slightly larger universe, add a few more edges. Maybe suddenly the robustness would really jump up. And then, okay, so maybe we should choose something larger. So I really kind of feel like I should answer all three questions in a quantitative way. And then depending on what you really care about, you would take the weighting and decide what you think should be a modular representation for that. Is that okay? Yes.
Behind you. Still stuck on the 0.6%. Since you mentioned you're only looking at the transitions in the data, so you're not using a lot of the data.
Right.
Is that why there's you dropped to only 0.6%? Because this is a very rough approximation you've got, right?
Yeah.
I mean, it looks effective, but it's an approximation.
Yeah.
You're getting so far, and then you stop.
Right.
After that, then maybe you use a completely different approach to whittle down that last piece.
Which, again, I would say that's why I really took the opportunity to come back and make that statement.
Yeah.
I think of this as a modeling framework,
Yeah.
but I know how to talk to differential equations.
Yeah.
And differential equations give us much, much more information.
Yeah, that's why you only get down to 0.6.
Yeah.
Or only
Right.
0.6. After that, you do as a
You
different
might
approach.
need, yes, right.
The only issue is the missing edges, I guess. If there are missing edges you're assuming your edges are correct. If there are missing edges.
But
Yeah.
again, the modeling framework computationally is a different question, right? But the modeling framework does say, oh, I could add more edges. I could add another node and
Yeah,
just
you could,
abstractly, and it's a framework in which to explore that very efficiently, right? I mean, we did 1 billion computations, right? And we can replicate those. You know, it takes a while, but on a cluster, it's
You're
not.
basically building a model ensemble.
Yeah. Yes.
A structural model ensemble, and then
Yep.
you're whittling away those who you can falsify.
Right,
effectively.
right. But I know how it fits. Again, it's not just sitting there, you know, on left field.
Yeah.
I know how the Boolean models fit into this.
Yeah.
And I know how to go back to differential equation models. And that's, to me, also
Yeah,
important.
no, you're using the Boolean models, but that's the easy way to get to this 0.6.
Yes, right.
Yes.
Except the dynamics that we're modeling is dynamics that I know transfers to differential equations.
Yeah, no, that's fine. You can then
Yeah.
transition afterwards. Yeah.
Right, right.
Okay.
All right, thank you very much. We're out of time because one should never put science above food. But I just have one comment. It's very interesting. Basically, what you're saying, Konstantin, is I take a piece of the cell's machinery, and I will not tell you sort of characterizing what a module is. I just tell you what it does or does not do. So it's a little bit like with gravity. We don't know what gravity is, but we sure know what it does.
Right.
But you have to have the players. You have to have the players. He has to have that.
Correct. Well, let's thank Konstantin again.